Hello friends, welcome once again in the video, and today we are discussing on the topic gram staining or gram staining of bacteria. The story starts with a patient. He goes to a doctor or registered medical practitioner. He explains the symptoms of the disease he is suffering from to the registered medical practitioner or the doctor. But the doctor is not able to prescribe an antibiotic because the registered medical practitioner or the doctor, he himself is confused that what kind of infection that the patient is having. So, in order to ensure the sample of the patient is taken and it has to undergo the different tests. Now, one of the tests is gram staining. Because the registered medical practitioner, as a matter of rule, he cannot prescribe any antibiotic blindly. So first he will confirm which kind of bacteria is present in the sputum or the patient is suffering from the infection, then only the treatment can be started. So we are discussing on the gram staining. Once the result of gram staining are there, then treatment can be started. So what does it mean? What is gram staining? So let us start. The gram staining, it is a technique to identify the bacteria in the sample. This technique was developed by a scientist or a histologist, Christian Gram, in 1884. It is a method of staining bacteria which is comprising of 4 steps. Means gram staining is done in 4 steps. What happens? Slide is taken. The sample is taken, smear is prepared. Now, this is the sample on a slide. The first step is primary staining. Staining means imparting a certain color to a sample. It means staining. Staining is an English word, you understand it. In the first step, primary staining is done. In the second step, another chemical is added that is called mordant dye. In the third step, a decolorizer or a chemical substance like alcohol is added. And in the fourth step, again a different dye is added, and this step is called as counter staining. Once the sample undergoes these steps, what is the result obtained? This result is of 2 types. Either the sample at the last, it appears like this, violet, and either in the second step or the second result is it appears as red. Now, on the basis of the result obtained after going through these steps, the bacteria are divided into 2 types, gram positive and gram negative. If the results are obtained like this, means if the bacteria are appearing like violet, these are called gram positive bacteria, and if the result is appearing like this, like red color, the bacteria are called gram negative bacteria. So what chemicals are being put in this test? How it is done? Let us come at this part. Here we will see what different chemicals are being put in this test. Now, these 4 steps are primary staining, mordant dye, alcohol, it is used as decolorizer, and counter stain, here it is counter stain. Sir, how to remember this? So we have developed one mnemonic. What we are doing? We are identifying the bacteria. How? We are decoding. So it is the mnemonic. It is means gram staining is primary method of decoding. Decoding of what? Bacteria. Because once a thing has been decoded, the actuality can be known. So means primary means this is primary. M means method, DE means decolorize, CO means coding, and bacteria is SS as we are doing. So primary staining, modern dye, decolorize, counter stain, these are the 4 steps. And for this we have developed mnemonic primary method to decode bacteria. So these are the 4 steps. Now what different chemicals or dyes are added in these 4 steps? Again we have developed one more mnemonic. What is that mnemonic? Crystal issues are always safe. Crystal issues are always safe. So crystal is crystal violet. I issues, I represents, I means iodine. All means alcohol or acetone. SAF means safranin. So this is part of mnemonic. Now let us understand the complete procedure. As we have seen, as I have told, the gram staining procedure, it contains of 4 steps. These are the 4 steps. Now what we do in each step, let us see. Primary staining method, this is the first method in which a dye like crystal violet or methyl violet or gentian violet is put onto the sample. So here is the primary dye and it is applied on the sample in the first step. Second step, mordant dye is applied. Now what is mordant dye? It is iodine solution which is dilute. So this is the second step where the iodine has been applied. Now this step is very important because the question has been asked many times in the exams that mordant dye used in the gram staining of bacteria is iodine. Now why iodine acid? Why it is added? We will see later on. We will first see third step. Third step is decolorization. Means in this step we are using alcohol or acetone. As we can see, alcohol, acetone or aniline is added or any organic solvent is added. It acts as decolorizer. So alcohol is acting as decolorizer. Now fourth step. Fourth step is counter stain. Counter means this is the step. And the counter staining is done by safranin, neutral red or carbolfuchsin. So mnemonic is this part and these are the different steps in which the dyes and the different chemicals are added. The questions have been asked from the each step or the different steps what kind of dye has been added at different steps. So don't forget, mnemonic is there to help you. Now let us understand what happens at each step. When it is the primary step, it is the first step, primary staining is done, means whenever crystal violet is added, the sample becomes violet in color or blue in color. After that, we are adding mordant, we are adding iodine. This iodine when enters into the sample, see what we are doing. We are staining the bacteria. Where bacteria is, it is in the sample. Whose sample? The patient sample about which the registered medical practitioner or the doctor wants to confirm that whether this bacteria is present or not. So once if the doctor is assured, he can start the treatment by prescribing a certain antibiotic. So this is the sample. This is the first step when the sample or the bacteria it appeared blue in color or violet in color. Now in the second step when iodine was added, the iodine enters into the bacteria and it helps in the binding of dye with the components of the bacterial cell form. Means presence of iodine, it ensures bonding of dye with the bacterial cellular components. This iodine also binds with dye and forms a complex that is called iodine dye complex. So this is the second step. Now in the third step, what happens is excess dye is washed with help of alcohol, acetone or any organic solvent. So if this decolorizing agent is added and this sample is washed, means everything will be gone out depending on the type of bacteria present here. In the fourth step, a different color dye is added, that is saffronin, and after that it is observed under the microscope. Now, the bacteria which is present in the sample, it may appear like violet, it may appear like red. If it appears like violet, it is gram positive, and if it appears like red, it is gram negative. Why it happens? So let us see. So after going through these steps, the two different kind of bacteria or two different type of results are obtained. Either some of the bacteria which are present in the sample, they resist decolorization. Decolorization means third step, they will resist. They will retain the primary stain. They will retain the violet color, and they appear violet like this, like this. The bacteria which resist decolorization, which retain the primary stain, and which appear violet, these are called gram-positive bacteria. So this is how we are going to confirm that the bacteria present is gram-positive bacteria. For example, there are many gram-positive bacteria. Examples are Mycococcus, Diplococcus, Streptococcus, Staphylococcus, Bacillus, Clostridium, Corynebacterium. Now, the second part of result. Out of sample, either this result may be obtained, or the second part is the bacteria may decolorize. Because of this, they are counterstained, means they will retain this color and they will appear red. It means after washing, the sample or the bacteria which are present in the sample, it will be decolorized, they are counterstained and it will appear red like this. If it happens, the bacteria are called gram-negative bacteria. And the examples of Escherichia, Salmonella, Vibrio, Neisseria, Shigella, Proteus, and Pseudomonas, and many more examples are there. So this is how we are going to confirm that the sample contains this or this, positive or negative. Now, sir, again and again you are telling gram positive or negative, positive or negative, but what we are getting in that? My dear, this gram and negative and gram positive, they differ from each other in many characteristics. For example, they differ from each other in terms of pathogenicity. They differ from each other in terms of their effects on the body. So, we are going to see the significance or the importance of gram staining. It is the method for identification of bacteria. It is a method which should be required and it should be done, that whether the sample contains gram positive or negative. Because this gram and positive negative bacteria, they differ from each other in terms of staining, as we have seen. They differ from each other in terms of structure. For example, the gram-positive bacteria has a thick cell wall. Cell wall is also known as peptidoglycan. So, gram-positive bacteria has thick peptidoglycan, while gram-negative has thin peptidoglycan. They both differ from each other in terms of growth requirements. They differ from each other in terms of pathogenicity. For example, gram-negative bacteria, it is more pathogenic. It secretes endotoxin, which is responsible for endotoxic shock. Details we will see in other videos. And they differ from each other in terms of susceptibility towards the antibiotic. Maybe the antibiotic prescribed by the doctor or the registered medical practitioner, it may not be active against the bacteria, because he didn't confirm that what kind of bacteria was present in the sample. But it can be confirmed gram positive or negative by gram staining. Now, why it happens that some bacteria appears violet or some appears red? We will see the reasons or the mechanisms behind. First mechanism, gram-positive bacteria, it has more acidic protoplasm and therefore it retains basic dyes more strongly as compared to gram-negative bacteria. Therefore, it appears like this. Means bonding between the protoplasm or the cellular components of the bacteria with crystal violet is more because of protoplasm is acidic and our dye is basic in nature. Second, the iodine and dye. This iodine which was added in the second step, which is also termed as modern dye, it forms a complex that is called iodine dye complex. Now as we have seen, gram negative and positive, they differ from each other in terms of structure and in terms of cell wall also. Now in gram positive bacteria, the cell wall is impermeable to this complex. The cell wall of gram positive bacteria is impermeable to iodine dye complex and it does not allow the outflow of this complex and it retains, therefore it appears violet in color. But the gram negative bacteria, the cell wall is thin and it allows the outflow of this iodine dye complex. Therefore, the primary staining method or the primary stain, it allows to wash and therefore when it is counter stained, it appears at red. So this is how the patient gets the right antibiotic and he becomes okay after the right antibiotic has been prescribed. But how the doctor has been confirmed? By gram staining. So gram staining, it is not only the one method by which the type of bacteria present in the sample can be ascertained. Different methods are also there, but gram staining method is also one of the important methods.